welcome, folks. Thanks for joining us at OpenHour. We're here with Max Lebron, and we're going to be talking about BabyLegs. But we're also some of us are also a nonprofit called Public Lab. If you are not familiar with us, we do a lot of open source community science, and some projects like BabyLegs and the work that Max does is near and dear to our hearts and our mission. This open hour and all of the notes and sort of all the stuff that you can sort of look at to sort of find out how to use your kits if you're a Kickstarter person, you'll be able to find that in the Public Lab website, discussion tag forums, all that stuff. So welcome. I see some names that are not familiar to me, so that's exciting. And I would love to just start off with some introductions. Maybe actually, Max, if we can sort of have you just very quickly introduce yourself and then give more of an introduction to the projects after we go around. And then let me start.
Sure. I'm Max Lebron. I'm the inventor of BabyLegs, and I'm working with Public Lab to sort of get her into more hands.
Great. And then the next person I see is Amanda.
Okay, hi everyone. My name is Amanda, and I work for Public Lab. I support the store and work on the kits team.
Okay. And then I see a car full of people, so we'll start left to right. Mimi.
Hello, I'm Mimi. I also work with Public Lab on their education team.
I'm Jeff. I'm driving, but I work at Public Lab as well. And I won't make eye contact very briefly, but but I've been involved in the BabyLegs campaign as well, making the video and stuff like that.
Hi, I'm Kay. I work at NASA Wallops on the Eastern Shore of Virginia. I work on a project called Erin where we use kites as a science platform.
Great. Then Liz.
Hi, I'm Liz Barry. I'm calling in from Brooklyn, New York, and I'm here to help this call run better. Thanks.
All right, and Michelle, I'm going to take you off mute. So if you want to say hello, or actually Liz, can you take Michelle off mute? Yeah.
Yes, Michelle is off mute.
So if you want to say hi, feel free. If not, we'll move on. Okay. Feel free to hop in anytime you want. Also, if you are not sort of able to talk, there's a chat button at the bottom that opens up a chat on the right side. So if you have questions you'd rather type in, that's also great. And we'll keep an eye on that too. And then D. Sarian.
Hi, I'm Sairam from India. I'm a community mapping fellow at Public Lab. Right now, I'm a research fellow in NIT Trichy in India.
Okay. Welcome. And Justin?
Hey, I'm Justin Schell. I run the Shapiro Design Lab here at the University of Michigan Library. I know a number of you from various edgy projects and other things like that, but I'm eagerly awaiting the BabyLegs to get and to show off here, and so excited to hear what everyone has to say today.
Great, welcome. And Fred?
Hi, this is Fred Blanton. I'm based in Stanton, Virginia, in the center of the Shenandoah Valley, western part of Virginia, and we won a grant from AGU, one of their 100 worldwide text projects, and it's a plastics waste project, and we're going to be measuring micro and macro plastic waste, and the scientist assigned to us is named Wynn Calger. He's a PhD student at UC, University of California in somewhere, I forgot now what town, and And so we were looking for something to use for the project, and then your BabyLegs project popped up. And so it was just perfect timing. So as soon as we get our kits, we're going to be using them as part of the APU text project with WinCaltry. Thanks.
Great, and I apologize, my internet sort of phased out for a second there, but yeah, I'm Bronwyn. I work with in Brooklyn, and I'm part of Public Lab's Kids Initiative, and yeah, just welcome everyone. Thanks for joining us, and I'm gonna pass it over to Max to talk a little bit more about the project.
Great. Don't forget to mute your mic if you're not talking. By the way. So I'm going to do a quick demo with BabyLegs about how to build her. It seems pretty straightforward, but the more I run workshops on her, the more people run into sort of the same snags over and over again, and they might not be intuitive to deal with. So I'm going to talk about some of those as well. I've built probably hundreds of BabyLegs, and because I invented her, I don't run into those snags. So it's always useful to talk to other folks. One of the really great parts about teaming up with Public Lab on this is that they have an online forum for all the users of BabyLegs, so people can talk to each other and troubleshoot, and I'm also very active on that platform. So if you start to use BabyLegs and run into something, you can always post there, and someone will usually answer you within 24 hours or so. So that's pretty good. It's a good user community. So I'm going to start with what BabyLegs can and cannot do. So the very basic anatomy of BabyLegs is she goes over some sort of body that keeps her open. It's a pair of tights. The tights are an extremely fine mesh. We haven't measured them yet. That's on the docket of things to do. It's one of the things that the Kickstarter is going to do. So some of you might know that there's a Kickstarter going on right now. There's about 48 hours left in it, and it is to perform validation and also some sort of top-down science on BabyLegs, validate her for various forums that care about different forms of validity. So plastics for the surface water, roughly half of all plastics sink, the other half float because of their relative density to water. Plastic, you keep the water level sort of here in the middle of the mouth, water goes in. Anything floating, including but not limited to plastics, collect in this, and now you've got this net. The scientific standard for this kind of work is called a manta trawl. It's about $3,500 US. I've got one sitting in parts up over there. It's heavy, it works really great, but it's also prohibitively expensive. So I invented BabyLegs before I had any grants. I've also got one other trawl. I'll show it to you here, I think. Sorry for dizziness. This trawl here is called the lady trawl. She's sort of halfway between BabyLegs and the manta trawl. She costs about $500 US, and most of that is the plankton net at the back. And so she's a lot more like the manta trawl and can do everything a manta trawl can, and she's been validated and all the rest. So they're sort of different levels depending on what you're trying to do. So what BabyLegs can do is collect plastics like this. So there's a couple of different sizes in here. Some things are very, very small. Some things are larger. Micro, meso, macro plastics, if you care about those designations. Maybe you don't. Here's another example. These ones are pretty small. These ones came from the Delaware River. And this is some of the smallest things she can collect. It's a trick because you actually can't see with your eyeballs what's in here. They're microfibers, like individual lints, basically, from synthetic clothing that usually come out of laundry, sewage. It's how you can tell that you've got sewage in your BabyLegs, is you come up with a lot of these microfibers from clothing. But she hasn't been validated yet, which means that I haven't put her in the water side by side by the manta trawl to make sure she collects exactly what the manta trawl does, which means that you can tell things like what's in the water, what are the relative ratios of different types of plastics in the water, so say styrofoam or foam plastics versus microbeads versus these sorts of things. But until we validate her, you aren't going to be able to say with total confidence, scientific style confidence, that what the density of plastics in the water are. Sometimes that doesn't matter, right? Especially if you're trying to do charismatic science for getting policy to do its own science, for example, but sometimes it does matter. What we will be doing this summer is validating BabyLegs. I anticipate that sometimes she gets exactly what the manta trawl does, probably in still water at low speeds. And in choppier and high-speed water, I think she skips on the water and gets very different data. So that's what we'll be working on telling for sure. Yeah, so I hope that helps. I think it's really important to talk about the limitations of the device, but also I've dropped her in the water a lot. And I don't use her for, say, public, like scientific publications, but I use her a lot to tell, say, where I might sample once I need to rent out a boat or something like that, because I can use BabyLegs with like a canoe or a paddleboard before I need to go rent out the $800 a day research vessel or something like that, or to tell where my hotspots might be so I can figure out my sampling regime or something like that. And I use her in schools and in community projects, and we actually don't need to figure out density for some of those. So that's what she can and cannot do. We can talk about this more afterwards if you want, but right now I'm going to do a quick as I can demo of how to build her. So I'm going to tilt this down here. So basically you can use any body you want for BabyLegs. Sorry, there we go. The body I'm going to use today is just a very small, very flimsy chocolate covered blueberry container. This is the kind of container that would do well in, say, a pond or some very still water, using it by hand, using it off a wharf. Would not do well in, say, choppy water or for behind a motorized boat. For that, I would get something like this. Much heavier, much more solid, right? Some chop isn't going to handle it, and it'll probably sink a little bit in the water. This is going to float in the water if you start going very fast and skim above the water and not collect a sample. You can always weigh it down, but this is solid and better. So depending on what you're going to use BabyLegs for, you would get a different body sort of suited to that. The sort of happy medium is like a cranberry juice container with like the ribbing in the middle. You'll notice that everything I showed you, both of them are square. That's mostly because if you do end up wanting to look at the volume of the water that you're looking at, doing math on a square is much easier than doing math on a circle in terms of calculating how much water might go through. It is also much, much, much more stable in the water. So, step one, you make it so that water can pass through the body of your container, which basically means taking off the top and the bottom of your container. The other reason I'm using a flimsy body is that I can avoid all the super embarrassing trying to cut through something thick issues on camera. There we go. The bottom's off. Now water can go right through. Now figuring out which is the front and which is the end, if you have a lip of any kind, which this one does, right there, while this side doesn't, I always put the tights on the side with the lip. So it's sort of like the plumber's clamp we're going to put on her acts like a belt. So it's sort of like if the water is trying to pull your pants down, you want to try and make sure that you've got a nice lip right there, right? If it's on this, see how hard that, it's actually hard to pull off. If I put it on this side, water can pull her pants right off. Not what we want. So, so this means that this is the front and this is the back. The other reason is that the front is slightly smaller than the back. For something like this, it wouldn't matter. But say you had like quite a tapered container. If your container is super tapered, you want to make sure that the narrow end is at the front, because otherwise your water might choke. You don't want your water to come out the front of your device again. I would highly recommend choosing something that doesn't have sort of a choke point inside of it. We've tested a bunch of different designs where say you're using like a container like this that's pretty long, or like a cranberry juice container, and we've scientifically shown that it doesn't quote unquote choke. Your sample always goes this way. It doesn't sort of get glommed up and pop out the other end again. So once you've got this situation, you then put holes in it. You put holes in it for attaching the rope to, which means the front. Now, I don't know if you can see this, but I've got 2 dots there and 1 dot there. I know it's hard to see. Sorry, reflection cameras. We've discovered that having holes in a triangle configuration makes things much more stable, where this is the top and this is the bottom. And so when you're going along in the water, if something happens and it starts going down or it starts going up, this is the configuration where you can sort of use the ropes like a marionette to control it better. 4 and 4 works for towing, but it doesn't work necessarily if you start like going like this or like that, right? So this 1 on the bottom and 2 on the top is the ideal marionette baby leg situation. Now at this stage, a lot of people start stabbing with an X-Acto knife. You can certainly do that, but I highly recommend a 1-hole punch. This one is also shaped like a flower, so it's also got this sort of beauty aesthetic going for it. And see how easy that is? No embarrassing stabby. If you do use the stabby technique, which is legit, just make sure that you burn off or around or otherwise sand the little holes. So you would like light that up. I'm indoors, so I'm not going to do that. Because otherwise you could get little tiny pieces of plastics around the hole that then end up in your sample and or in the ocean, neither of which we want. We've got these nice beautiful holes, 2 on the top, 1 on the bottom. And now we can put on her pants, get her dressed. So the tights are the most expensive part of baby legs, for perhaps obvious reasons. Now these tights need to be nylon. They cannot be cotton. Cotton sinks. And then you'll end up in a situation where like your baby leg starts to drown. This is, I underestimated the like puppeteering effect that this demo would have. It's great. So you want to make sure it's nylon. And we tend to make sure that it's pink, ideally hot pink, although this is sort of a light pink. And the reason for using colored tights instead of white tights is you can tell if your sample is contaminated by the nylons from the tights themselves, because very few plastics in the quote unquote natural environment are pink, right? They tend to be white, black, blue, clear, that sort of range. So pink is for science, not just for hotness, although both are achieved. So next you put on a plumber's clamp. This plumber's clamp, you can get ones with little wings on them that you twist. This one you can use a nut driver on or a screwdriver. This is a nut driver. If you're going to be doing lots of sampling in the field, I highly recommend a nut driver instead of a screwdriver, because this is a knuckle buster, right? So you'll be slipping, your hands are cold. I do a lot of work in the subarctic, and so when my hands are really frickin' cold, this device works way better than other devices. Nut driver. Sounds dirty, is a little bit, but very effective. Now before I get into more on the plumber's clamp, let's say you have this situation where, oh no, your plumber clamp is too small. Well, did you know that you can join 2 plumber clamps together to make 1 bigger plumber clamp? Most people who aren't plumbers don't know that. You can. You just join them together on the edges, screw them in. What if you have this situation? Oh no, my plumber's clamp is way too enormous. This is actually really great, because if you do a baby's legs like this and then bend this down, well, first of all, baby legs now, and this is down. This goes down into the water. It weights baby legs. This is now hard to show. It weights baby legs and also acts as a sort of rudder and makes her a little bit more stable. So this scenario isn't bad at all. In fact, it's good, especially if a really light body. It does make baby legs look like she has a penis, but it's a very functional penis. So that's good. I prefer the tight fit, mostly just because of aesthetics. Now, if you've followed my advice and gotten a square body, which is more stable than a round body, you're gonna have to do a little bit of crimping work to turn a round plumber's clamp into a square-ish plumber's clamp. And that'll keep the baby legs on a little bit more securely. So it is kind of like dressing a baby, a little bit frustrating, but you can be a little less gentle. All right, where'd my nut driver go? So there's the nut driver. Tighten it up. This is also way faster than a screwdriver. All right, so see how she's sort of, this is a very thin body, so she's sort of squishing. It's a little bit tight, so I can undo her, put it down at the very end, and screw it back in. It doesn't matter if the mouth isn't perfectly formed. What matters is if her tight falls off. So the test is you give this a really good yank, because water is a lot stronger than you think it is, a lot stronger than you think it is. And so if you give it a lot of really good tanks, maybe play a little gentle tug of war with it, that's your test. Sometimes I'll take the edges of the tights and fold them over and then screw it in a little bit tighter, especially if I'm going off a motorized boat. Sometimes I'll put 2 plumber clamps if I'm off a motorized boat, because again, that gets really strong, especially as BabyLegs fills up with organic material and plastics. But for something that's this small, this is fine. So there we go. She's got legs. Excellent. Let's give her some rope. So I have color coordinated my BabyLegs today. That's not necessary. Doesn't improve the signs, but it does improve the general look. So one of the things that's important with BabyLegs, with putting the string on, is to make sure that the rope doesn't have a bunch of microfibers that 1 might go into the water or 2 end up in your sample. So you can burn it. Again, here's my little butane fire starter. But if you're indoors like I am, or if you're working with kids where maybe fumes aren't a good idea, you can use a number of types of tape. This is science labeling tape. And just tape off the end. This will keep microfibers from going in the water, and also you can point it up so that it gets really easy to go through that. All right, now, how much do I want here? For a little one, maybe this much. All right, so, hard for me to tie and talk at the same time. So what you're going to do is you're going to put 3 lines in. What you'll see is that this is all 1 line. I don't do 3 individual cuts. I let the top be essentially the same line. The less cutting, the better, only because it means less potential microfibers running around. All right, make sure these are about the same distance. All right, and then 1 last cut. It's like watching paint dry, but different. All right. The steps I just showed you, by the way, take new people like 20 minutes. So I'm feeling quite accomplished, even though that took a while. All right. So if you know how to do a fancy knot, because you're like a fancy boat person or a fancy fisher person, that's awesome. But a square knot, which is what most people just call a knot, works just fine. But again, make sure you either melt or tape off the end of it. All right, that's another reason just to leave this. Okay, so this is the bottom now. Oops. All right, so what you do, this is hard to show. This is very hard to show. Okay, so what I'm gonna do is I'm gonna try and tie off the top and make sure that my BabyLegs is totally straight, not tilted like this, not tilted like that, but basically tie it off. If you have another person to hold BabyLegs steady, that makes it even better. I'm going to tie it off, and I'm going to leave, oops, at least 1 loop at the top, where I can attach her to a longer rope to tow behind a boat or something like that. Oh, this isn't working. There we go. There we go. This will work. And then tie it off again. If you can do a bowline, the bowline, especially if you're going to put it behind a motorized boat, the bowline is the best knot to use. If you are used to boats and stuff, you'll already know that. If you hang out with boat people, they'll tell you. But see, basically at the end of the rope, I have this loop now. So this is BabyLegs. And on this rope, this is my weight-bearing knot. So this has to be my best knot. This is where you'd put a bowline or a double knot or a triple knot or something. melt it, glue it, whatever, make it strong because this is your weight-bearing one. And this is where I would attach my tow line. And your tow line can always get changed out and be variable. If you are working in a motorized situation, I usually put a C-clamp there because then you can just attach and unattach, which is a little bit safer and a little bit faster. But if you're doing hand tows, it doesn't matter. So when this is in the water, so there you go, BabyLegs done. Ta-da. Easy peasy. Takes a lot of people about half an hour to an hour to do their first one. I make these like once a week. So I'm going to touch a couple things about putting it in the water. There's all sorts of ways you can put BabyLegs in the water. You can tow her behind a boat that's either human-powered or motorized. You can drop her off a bridge and just leave her. You can do it off a wharf. You can do it by hand with some boots on. You can do it on a paddleboard. You can put it in flowing water and just hold it, right? The exactitude of what you use depends on what kind of science you want out of it, what kind of knowledge, whether you need to know how much water is passing through or not. But she's pretty versatile, and she can handle about 5 knots on a boat, which is quite fast, as long as there's not a lot of chop. Yes, people who are talking about my dogs. This one's Kookum. Oh, the one who's bothering me is Kookum. The one that's not bothering me is Grandmother. They are lap dogs. They have a special contamination jar with their fur in it in case they shed and get in our samples, so we can tell. That's right. So the first thing that tends to happen when this hits the water is that she either floats above the water and won't sample, or like starts sort of going nuts and twirling. If she's twirling, it's because of where your holes are. You can adjust that by adjusting the string and maybe changing where the holes are, making them farther apart, making them more even. And if she's above the water, it's because you're going too fast and or your body's too light. So that's where you would use one of these bodies. Perhaps you've seen, sort of super duper versions like this one, right? These are flotation. These are for like when you're going really, really fast. On a motorized boat, and the flotation is actually to keep it up in the water, but you can also put rocks or sand in these to weight it down as well, in little pontoons that you can just zip tie to the sides of your BabyLegs. Or you can just sort of adjust your speed. Yeah, those are the main troubleshooting things. Once you get her out of the water, you get to just look at your sample like you're turning a sock inside out, and your sample's right there. Again, depending on what you need the sample for, you can either rinse the whole thing into a sample bag or a sample jar. If you do that and save it for later, I highly recommend you use some hydrogen peroxide, because otherwise you'll start to smell like death, because things will be dying, organic things. If you're doing lots of sampling in the same place, I highly recommend bringing many, many tights and just putting them in Ziplocs and labeling them, and having a sheet where you label too, because getting the sample out of BabyLegs, things sort of get into the weave, and so you sort of need a lot of time with some running water to get things out again, and some tweezers, and maybe a microscope, while just switching out the tights and putting them in bags. That's what we do. It's faster, it's kinder, you get a better, more reliable sample. So that's the skinny on BabyLegs. There are some questions in the chatty thing that I'm going to start on, and then we can also just unmute if we want to. Many comments about dogs. That is typical. So if you're trawling with BabyLegs behind a boat, what are the signs that you're going too fast or you're about to lose her? We have never lost a BabyLegs, actually. Mostly because I'm paranoid about losing the tights, so they're pretty secure. If you're going too fast, she'll either skim above the water or start to skip. That means you're going too fast. If she starts vibrating and shaking, you probably want to slow down. And if any point you see movement on your plumber's clamp, you should slow down. Yeah, if I'm behind a boat, again, I double clamp, I make sure there's a ridge, I make it really tight, I fold over. And like I said, 5 knots is pretty fast. It's what you would do a normal manta trawl at, or a lady trawl, and we don't lose them. Yeah, never lost. That being said, it's much more likely that you lose the entire BabyLegs and not just the tights. And so when I'm on a boat, I also attach a leash on all my trawls, not just BabyLegs. Basically another hole that just independently goes to the boat. So if something goes sideways with this, you don't add more pollution to the water by losing your science object in the water. Yeah. Anything else? Questions, comments, concerns, thoughts?
Well, I really enjoyed the presentation. That was so clear and understandable, and you're also a very entertaining presenter, so thank you so much for coming on. I loved being able to watch you actually assemble it. And yeah, I mean, we could officially open the floor to questions from those of us who are here in the video call. And then I'm also keeping an eye on the chat room in case somebody types in there. Something else, now that you're listening to me, I'm going to share one link in the chat, which is to where we've been taking notes. So if there's something that you appreciated, that you learned, that you wanted to jot down, or wanted to make sure that, you know, other people would also notice who came later, please toss it on in their notes. And
so one of the questions that just popped up in the chat from Mimi and or Jeff is, how reusable are the tights? The answer is very. So the other reason we use toddler tights instead of women tights is that they're very robust. I don't know why women don't have funner, more robust tights, but they don't. They like, what is it called? Run very easily, and toddler tights don't, I guess because crawling. So I will reuse these many times and just throw them in the washing machine, honestly, between things, especially if I'm trawling in sewage infested water, which I very often am. I don't wash it by hand. I just put it in the washing machine with vinegar and grossness. And so basically I will keep using her until either there are holes or it starts pilling, and then I worry about contamination. So depending on how rough you use them, 4, 5, 6 times. Yep. Question also, how to handle or prevent collecting sea life. So you may or may not know this, but if you're a scientist and you ever do surface trawling, you always have to get a permit because you are, whether you want to or not, trawling for plankton. Plankton are very small. They will also get caught in BabyLegs, so will krill, bugs, little tiny fish, all of which have been caught by BabyLegs before. When I go through and I pop this open, if anything is still moving, alive, I take it out, give it a little rinse to make sure that if there's a sample stuck to it, which sometimes there is, it gets a rinse and then plopped back into the water. But things are going to die, things you can't even see. That's what putting the peroxide into the sample actually does. They will die whether you want to or not, and they will smell like death. So the thing for like larger fishes, like vertebrae and stuff, they've survived however long, not falling into the open mouth of things like BabyLegs, they'll be fine. On a high-speed trawl like the manta trawl, they do kill animals because the pressure in the sock. Not so with BabyLegs. It doesn't go. She just can't go that fast. She doesn't exert that kind of pressure. Jeff, are plankton edible? Delicious. I have actually tried to find plankton recipes before because our lab has a rule that you have to eat all our samples, and there aren't any. I think they would be salty, the ones that live in brackish water. Otherwise, I don't know. There's a question here about when the Kickstarter kits will come out. Someone from Public Lab, you can answer that way better than I can because you are the disseminators of the goodies.
This is Amanda from Public Lab. I can help answer that. I believe if you're talking about the $90 or the $125 reward level, the $90, the BabyLegs globe and microscope and poster kit are scheduled to ship in September. And the kit with the plus microscope, so the upgraded microscope, is slated to ship in October.
The good news is that the kit just makes this easier. You can do all of this without the kit. And people have been for a really long time. The kit is really just to sort of give it that much more ease, people who can't get to stores or whatever. So, yeah. Like I just pulled this together based on, well, except for the tights, based on what was in my house and lab. So.
And Fred, there's no other questions in the queue right now. So if you would like to ask any questions. I mean, maybe you need to get your project going this summer, you know, before October. You can join us, just unmute and jump in. Oh, okay, great. Thanks for writing in the chat. I'll read it. For the purpose of the recording, I'll read it out. Fred says, I was just thinking that. Seems like we really can just make these kits ourselves. We'll just do that to get things going sooner. Thanks.
Yeah, the one bottleneck will be the microscope, and that's for analysis. But if you're working with Win, who I actually am currently working with too, he'll have other fancy scientific methods for doing analysis. You might also want to talk to Win about what kind of data you're trying to accomplish and what BabyLegs can and cannot do for that data. Again, until we have the density capacity validated. We have one set of data from California, but it's a really variable data set because of how the water was. And so it's not, that seems to indicate that in even water, BabyLegs does very similarly, statistically similarly to the manta trawl, but I want to do another test altering water state because I don't think it tells us enough reliably yet. And it needs to be published so that when people try and use BabyLegs data in like policy circles or something, they can point to precedent, they can point to scientifically published peer-reviewed sort of okayness. What was the most interesting you've ever caught? Plastics, because I'm a plastic geek, I find very interesting. Urban whitefish, which are condoms, some might find interesting. I don't know. I found it sort of warm. And then on another scientific sort of thing, we also caught a butt plug once, which I didn't know floated. And I actually had to teach my students what it was, which was interesting. Oh, actually, another really interesting thing we've caught, they look like little Advils, and they have a little pinprick in the top, and we couldn't tell what they were. We thought they might have been for like baiting fish traps, which is a big thing around here, but they're actually a type of pill where the only delivery system is that you have to encase the powder in plastic, and then you eat the plastic, and then you poop it out, and so it was an indication of sewage plastic. I thought that was pretty interesting, and we had to go to Twitter to help figure out what it actually was. A pharmacist came on and told us. So that was pretty cool. And now we know it's sewage plastic, not fishing plastic. Can you talk a little bit about the kinds of things folks are likely to find in different bodies of water? Absolutely. So all plastic pollution is place-based, characteristic of different places, characterized by water and land use. So in Newfoundland and Labrador, where I work, we tend to get a lot of fishing plastic. So like little green, what are called threads from fishing line, fishing gear, and sewage plastics, because most of our sewage, the vast majority of our sewage isn't treated. So sewage plastics are characterized by condoms, urban whitefish, tampon applicators, also called beach whistles, corn if it's fresh, microbeads from cosmetics, and microfibers from laundry, and poop if it's fresh. And toilet paper. So that sort of combination of things, in any sort of combination, will tell you that you've hit a sewage outflow. The Great Lakes tend to be characterized by a lot of sewage plastics, particularly microbeads, but also pre-production pellets. So little flat-ish, they look like beads. They're from industry. If you wanted to make something out of plastic, they would ship you a container full of these little beads. You would melt them down. Yeah, fishing gear. If you are on the, say, the West Coast or an area that gets a lot of come from away plastics, like the global swirl, basically out of gyres, you'll get a lot of fragments. Yeah, so those are, so depending on where you are, if you're in an urban area, you'll get larger plastics. If you're in a rural area, you'll tend to get smaller plastics, especially if they're sort of going through. You will rarely get film plastic, like plastic bags, mostly because it shreds into nothing immediately. Also, urban areas tend to have a lot of foam plastics. So all those things will tell you, so New York City, the #2 type of plastic that we find there are foam plastics from styrofoam, that sort of stuff. So, so you might not know when you go to a body of water what it'll characterize. What you find will help you figure out some of the potential sources. Is there a standard data reporting format for these samples, like a spreadsheet with standard columns to fill in? Yes, and actually, I don't know if I've shared one of those yet on the public lab pages. If I haven't, if one of you public labs could poke me and I will share it. It's color coded, it's lovely. It has things like longitude in, longitude out, which you can do with your phone, GPS, or a fancy thing. And then we also have an analysis sheet for the types of things. So the size, the weight, the types of plastic, and has metadata describing each one. Yeah. And so when I analyze, basically I'll give you the sheet that I use when I analyze plastics for other people. Yeah. The other thing is that using these sheets can go towards helping you characterize what kind of plastics you do. What we'll do in our lab also is do stats on them. Is there a significant difference between the number of film plastics and the number of foam plastics, for instance? If there isn't, doesn't really tell us much. If there is, it can start telling us about what the sources around here might be. So we also do stats. Sorry, I'm looking down the thing. Standard sheet, different bodies of water. As a follow-up to the question about standard reporting format for data, is there a place where everyone shares and compares their microplastic data? No, it's an ongoing issue. In fact, it's a huge issue in the science community that people use different things when they do analysis, which makes comparison really hard. That's actually what I'm specifically working on with Fred, is coming up with what you have to report to figure out how we can make our data comparable. I would say that the vast majority, probably 80-ish% of publications on plastics are in the Marine Pollution Bulletin, which is one particular not open source journal. The nice thing about it though is that if you email the corresponding author, they will almost always send you their paper, and they're legally allowed to. And increasingly, a lot of NGOs and scientists are going different places in monitoring, so you can always sort of use other papers as a comparison, not only to that area, but to similar areas. So we compare our plastics, for instance, to the North Sea, and the Baltic North Sea a lot, because it's quite similar to ours, for instance. Nurdles, question mark. Nurdles are the super cute surfer name given to, like literally by surfers in California in the 90s, given to these little beads that washed up, the pre-production pellets. They were like called Nurdles, dude. And it's still my preferred term, actually. And they're just pre-production pellets, exactly the same. Those tend to be found in areas with high shipping. High shipping. That's normally where you'll find a disproportionate number of Nurdles, because or high industry. So either they'll flow right out of industry, especially on loading docks. They'll come out, they'll get spilled out of container ships. And then there are some amount sort of in the giant global gyre sort of flow of things. But say around here, I almost never, ever, ever find Nurdles, because most of our water comes down from the Arctic. Never, ever. So that'll help characterize your area as well. And also, actually, in the Toronto area, scientists looking at Nurdles just managed to get an industry that was illegally dumping their Nurdles into the water fined because of this kind of research. And they didn't do a density. They didn't do density. They just sort of sampled in a bunch of areas, helped them pinpoint where the most were, and then found the industry. So that's pretty cool. That was in Toronto, Canada. Our creeks and streams are significant because they're the headwaters of the Chesapeake Bay. We're trying to ensure our water is clean before it gets to the bay. So yeah, in that case, you might want to sample all the way down the stream to find out if there's a switch that gets flipped, which would help you find the source of where plastics were entering into the water. The problem with plastics is they're not usually a point source of pollution. They're usually like broadcast off of most of them come from land. So that's always tricky, but that's how you would do a sampling regime. So you should also work on a sampling regime that tells you how to do what you're trying to do. Does velocity or wind speed affect BabyLegs? If so, how to ensure the plastics collected doesn't go back into the water? So velocity and wind speed do matter to the functioning of BabyLegs and where she is in the water, but not what she gets collected. So it'll affect things like chop and whether she's riding really hard and whether a bunch of sample is like splashing or like not quite making it, or like if she's constantly sort of dipping her head underwater. If water goes over top of BabyLegs, because plastics are in the top bit of water, you're losing sample. So you always want the surface of the water either halfway or 2/3 up If it's really shallow, then you're missing, so plastics are in like this much, basically, of the top of water, sort of churning around. When we do density, which you cannot yet do with BabyLegs, we just use it as surface area, not volume, mostly because you can never know the volume as water is going like this, truly. So you just calculate surface area based on the assumption that everything's sort of floating at the top. So that's what wind speed and velocity will affect. You should always be moving forward with BabyLegs. As soon as you stop moving forward, if any of the sample moves out, you're in trouble because your sample will float out. The nice thing about BabyLegs is that the legs crumple and eliminate some of that. It mitigates, I should say, some of that. But always you should be moving forward. And so that's why always having someone on the BabyLegs paying attention. So when you stop to slow down, just flip the head up. On our bigger trawls, we just flip the head up and put the water here. And then it doesn't matter how slow you go or if it starts going backwards because you're no longer collecting any sample. So that's sort of a way, you know, it doesn't matter if it goes back and forth because nothing's going in here or leaving here. So, yeah. Oh, also you should, when you're collecting your data, and this will be on the sheets, you do put down like the Beaufort sea state, the velocity, that sort of stuff that you're going on in case it affects your sample, because you can always test statistically to see whether it is affecting your data, which it shouldn't, but what it might be doing is misperforming your BabyLegs or your trawl, making it ride too high or skip or splash or whatever. Is there a size range that makes something microplastic versus macro? Yes. Okay, so there's macro, meso, micro. And there's some variation on this. Micro is up to 5 millimeters. We're in metric world because science. 5 millimeters to 2.5 millimeters is usually considered meso, although there's some variation on this. And then bigger than that is macro. So this is full of meso and a little bit of macro and a bunch of micro. This is probably mostly micro with a little bit of meso, no macro. Plastics get down to the nano size. Science is fine. There's a article by Song et al. That shows that reliably you can only go down to 1 millimeter, which is about that big, with your eyeballs and a microscope to reliably tell plastics from non-plastics. That is the scale at which my lab tends to cut off. After that, you need a spectrometer to identify plastics from non-plastics because you just can't do it even with a microscope. There are different techniques to do that. You start moving into science lab and out of citizen science land because it starts to get expensive and really technical. So about 1 millimeter is what you can reliably do citizen science eyeball style. And that's what we do so that our science is replicable by citizen scientists. It was a fad to feed styrofoam to mealworms recently. Is there a sea creature that eats plastics? Many animals eat plastic, much like mealworms. Then they poop them out, or not. They do not tend to break down the plastics into their constituent parts. There are some, like mushrooms and microorganisms, that do, but they don't really do it to scale. And the reason you know that is that we're not in the middle of a B movie with like little microorganisms eating our airplanes and our dentures and our bridges. So the conditions under which those things will eat plastic and change them out of their polymer structure into monomer structures is that usually they're starving and the plastic has to be covered with the bug equivalent of sugar. Over time, so there are other polymers in the world that aren't plastics: fingernails, toenails, hair, shells, right? And that, yeah, that's a polymer. But microorganisms have co-evolved with them and they do eat them. Plastic just evolved very quickly without its sort of rest of its ecosystem evolving with it. Eventually, in geologic time, after the human species has left, not because we're all doomed, but because just geologic time works that way, they will eventually get eaten. But at that point, environmentalism will no longer exist. So, but you generally probably don't want microorganisms or mealworms or whatever randomly eating plastic because so much of our infrastructure is plastic, and we would be in the middle of a B movie. So, FYI.
Just a quick time check. I know we've still got some questions on the list. It's 5:22, so we've got about another, 5-10 minutes on the call and a few more questions. But if there's any sort of burning last questions, stuff that folks want to sort of say about their work or ask before we hop off, now's the time to get it in the queue.
So there's a question about what a platform for sharing would have. That is actually something that Wynn and I and many others are working on. So that'll be out soon and we can share it. Also, the spreadsheet I have will be the basics for something like BabyLegs. There's other types of monitoring that involves other things. The problem is always sampling design. So what Fred asks about, like, okay, what if you have a moving creek and you just want to sort of leave a BabyLegs flowing in there, versus if you want, if you do it by boat, versus things start to get a little bit not as comparable in those cases. But so that's something that you would need a lot of metadata on and description on, but that's very doable. So Fred asks about, like, can you just leave BabyLegs like tied up on a stake or on a branch or on a bridge and just leave her in the water? Yes and no. I would eyeball it and stick around for it. So we had a trawl once. It was the lady trawl, the sort of bigger than BabyLegs, smaller than Manta. And we were tootling along, not too fast, and a dead seagull was in the water, and it went through, and we were going fast enough, and it was so heavy that it punched out the back of the trawl and broke the trawl. And then, of course, we lost our sample, but not the trawl because it had a leash on it. Also, we've been in a trawl situation where, so in the middle of the ocean, you can trawl for literally hours and it won't fill out your trawl. But if there's sort of like a rain event, or it's like there's a bunch of algae in the water, seconds, it can fill up in seconds. And then if you fill up the top, you no longer have a viable sample. So I would get to know the creek a bit and find out what its limitations are. Does it dump? Does BabyLegs end up twisting and then you don't know if you have a true sample or not? That sort of stuff. Eyeball it in a calm, not a lot of plastics, not a lot of biota sort of stream. You might be able to leave it overnight. I know we have in the open ocean left trawls out on the boat overnight with the Sentinel. But yeah, so it depends on your body of water. I would watch it first and then find out. Yeah, streams and rivers tend to have less biota than oceans and streams and ponds, so that actually might be a good case scenario, but again, you'd have to make sure that you had a real stable BabyLegs. And what a coincidence, we're towards the end and there are no more questions. It's like it was planned or something. What an organized group.
Yeah, that's amazing. Yeah, I mean, we can sort of hang out for a few more minutes and chat, but just want to kind of remind folks that we'll post this video and we will share out some notes, but you can also always come back to Public Lab and ask questions or talk about, your projects, whatever it is that you're doing, and there will be people to talk to about those. So, we're all collectively here for each other in this work. So there will be more chances.
And once we have the validation, it'll go on the Public Lab site. Basically, where I'm putting all of my resources now is the Public Lab site. Updates, all that sort of stuff is where the, whatever. So check back in a week, a month, 2 years, 50 years, that's where it'll be.
publiclab.org/babylegs.
Yes, I was just typing in that publiclab.org/tag/babylegs is where you can subscribe. So every time Max or anyone else posts something new about BabyLegs, you'll get an email, but only about BabyLegs. You won't get emails you don't want.
Yeah, and that's how I know when a user has an issue, and then I try to pop on and answer it. That's the same method. So yeah. Thank you very much, Public Lab. Thank you, everyone, for all of your thank yous. Thank you, Sexy Pants BabyLegs.
Thank you, Sexy Pants. Yeah, great, everybody. Thanks again for coming, and we'll kind of wrap up this call. But yeah, find us online and talk to us about BabyLegs and your other environmental issues, questions, all that.
And if you have an inkling to email me personally, put it on the Public Lab instead. First of all, because I might have already answered your questions, but also because other people will ask the exact same question.
Oh yeah, public science. Thanks so much, Dr. Max.
My pleasure. Thank you guys very much.
All right.
Bye, everybody.
So long, everyone.
Bye, y'all.